This is a production of Cornell University.
Thank you, Claire. So today I'm going to be presenting on my postdoctoral fellowship, which is still in progress. The title of today's talk is A Novel Socio-Ecological Approach for Promoting Area-Wide Adoption of Organic Farming Practices that Enhance Microbially Mediated Pest Control. So today we're going to be talking a lot about organic farming practices. And so the organic label or organic certification regulates practices that support various factors within farming systems. So some of the factors that I'm interested in are the social dynamics that occur within farming systems, the economic aspects of organic farming, and then of course the ecological aspects. And I'm very interested in the connection between the social aspects of sustainability within organic farming systems and then the ecological outcomes. So one of the ecological outcomes that we're interested in organic farming systems is microbiome conservation. And that's the reason that I came to Claire's lab. And so Claire found that organic farms help to promote microbiome diversity over conventional farms. And then Claire was able to follow this benefit of the microbiome up through plant defenses. So this is a plot with salicylic acid here showing that organic farms had higher levels of plants grown with soil from organic farms, had higher levels of salicylic acid. And then finally, Claire and her co-authors were able to track that all the way out through pest reproduction, where organic farms, the soils from those farms suppressed pest populations. And so the idea here is that organic farms help to conserve a certain aspect of the microbiome that promotes plant defenses and suppresses pest populations. So where my kind of novel wrinkle in this research comes through is that generally we look at organic farming as kind of this one big system or one big idea, but we know just by looking at Claire's data and then also data that I've produced from other research that there's a lot of variation within organic farming systems. There's variation across those farms in terms of their capacity. I'm sorry, I just saw something on this computer. Variation across organic farms in terms of their capacity to promote the microbiome. And then from my research, also the levels of pests vary considerably across organic farms. So one can look at this and think that the ecological benefits of organic farming vary. And one of the reasons that the ecological benefits of organic farming vary is within that label, organic farmers can use a spectrum of practices. So if you go out and you walk around on any organic farm in New York State, they're going to be using different practices and they're not just one homogeneous unit. So for example, some of my PhD research looked at plant diversity within organic farming systems and how that impacted wild bee communities. And so as plant diversity increases across organic farms, we see increases in ecosystem services like pollinator visitation to crops. And so what we assume is kind of underlying this variation in pest populations might be something similar, where organic farmers are using a spectrum of practices and that ultimately influences pest populations. So if we're gonna kind of spin off that idea a little bit further, we propose that this variation in practices not only mediates, for example, pollinator communities, but also microbiome mediated ecosystem services. So we're saying that what organic farmers do and how that varies across their farms ultimately might be impacting the microbiome and microbiome's capacity to regulate pest populations. So then taking a step back from that, I wanted to ask myself, why do organic farming practices vary? So this is kind of like a why question, right? So you're like, well, if organic farming practices vary and farmers are doing different things, why are they doing different things? And then the second question that comes out of that is, once we know about this variation and why farmers are doing different things, can that variation be leveraged to promote ecosystem services like pest suppression that we think is beneficial? So one of the reasons that there might be variation in farming practices across organic farms might be due to farmers' beliefs. So this is starting to step into the social aspect of my research. And I've shown in my research, as well as other researchers, that if a farmer, for example, believes that their practices are impacting pollinator populations, so they're like proponents of pollinators, like you see over here, they intend to engage in pollinator conservation efforts. So this is showing that what someone thinks about what they're doing ultimately can drive changes in their practices to promote conservation efforts. In addition to that, we think that there are a lot of reasons farming practices vary, and some of those could be related to their time in organic management. This is some data from my PhD work that showed that over time in organic farming systems, that mediates the assembly mechanism of pollinator communities. So you can kind of think about time in organic management as a proxy for different changes and practices that are occurring over time within those farming systems. And then you can also look at other researchers work on this topic and find that factors in terms of like farmer demographics, such as their income, can influence better management practices, like how they manage the soil. So it's important to say that not only is what someone's thinking or their beliefs mediating what they're doing, but there are also a lot of other external factors or factors occurring within the farming systems that can also influence the farming practices that they adopt. So taking this one level larger is that what we've talked about so far is sort of like this variation within organic farming systems. But what we wanted to do for this project is scale this process up. So could we take what we saw from Claire's study and scale it up to an area-wide program to promote the conservation of the microbiome, enhance microbiome-mediated ecosystem services like pest suppression. And one of the things that we've come to realize with area-wide management is it requires coordination and coordinated adoption, especially when it comes to suppressing pest populations. So that's something that we're looking at in this study as well. And that's something that my co-advisor on this project, Dr. Shadia Talla, is helping with. So this is kind of one of the general framing questions for the study is, can socio-ecological models study adoption and ecological outcomes at broad geographic scales? And I want to go ahead and introduce what socio-ecological models are, and I think I've already kind of alluded to this. So the idea is that what someone believes influences the practices that they use. So that's the first part of the socio-ecological model. And then through someone's beliefs that influence their practices, that then mediates ecosystem services. So this is the part of the research that I have focused on a lot of my career, which is just evaluating how farming practices influence an ecosystem service within a farming system. And this is the part of the model that I've also done before, which is just looking at how beliefs influence practices. But for this study, I wanted to create a full socio-ecological model, which would evaluate what someone believes and how that influences their farming practices. So what a farmer believes and how that influences their farming practices, and then the ecological outcomes all within a united model. And in addition to that, this is called a static socio-ecological model. But as part of the project, we're looking at how what we can determine in terms of the ecological outcomes in their relation to farming practices, how that information can be extended back to farmers and ultimately change their beliefs and shape the adoption of farming practices to help support ecosystem services on farms. So far I've used a pollinator as a model, but you can apply this to any ecological outcome and really any type of farming system that you might be interested in. And so for this study, what we're looking at is how farmer beliefs mediate their farming practices, like pesticide use, for example, and how those farming practices ultimately mediate ecological outcomes in the microbiome, plant defenses, and pest suppression. and then how learning about that knowledge can be extended back to farmers to, again, mediate and shape the practices that they're using to promote these types of ecosystem services. So this is the premise of my USDA postdoctoral fellowship. Again, I'm working with Dr. Claire Castile on this, but I'm also working with Dr. Shadi Attal at the University of Illinois, who is a social scientist and economist and does this type of modeling. So we're coming at it where Claire has been bringing in kind of the technical tools to evaluate the microbiome and plant defenses, and Shadi has been helping a lot with the modeling approaches. So today I'm going to demonstrate this part of the model first, which is how does what a farmer believe influence what they do, so their farming practices. And then I'm also going to start showing you some of the ecological results. So I'm going to show you the ecological results for the microbiome. And then I'm also going to show you the ecological results for pest suppression. So the idea here is at the end of the lecture, you'll see in a unified model how what someone believes can ultimately result in ecological outcomes within organic farming. So the first part, which is going to relate what farmers believe to their farming practices that they adopt, is a paper that that is now in review at People and Nature. And the title of that is Motivating Organic Farmers to Adopt Practices that Support the Pest Suppressive Microbiome Relies on Understanding Their Beliefs. So I'll go ahead and get into the methods for this paper. So we started with site selection through what's called the USDA Integrity Database. It's a broad database of organic farmers. There's about 16,000 organic farmers in this database. And the value of these farming systems is around $4.2 billion annually. And then as we narrowed our selection to New York, there's about 1,500 organic farms in New York State, and it's about a $216 million business annually. So some additional filters on our site selection. We only wanted to work with vegetable farmers, small fruit and vegetable farmers. So that narrowed our selection down to about 280 farms or so. And then I sent this list of farmers out to Cornell Cooperative Extension, and they were able to tell me if the farmers met my selection criteria. Okay, so as you might know or have inferred, that a lot of the data that I collect comes through survey instruments. So to evaluate what someone believes or what they are thinking, you have to ask them through a survey tool. So I'm going to talk a lot about the survey tool, but I also brought a copy of the survey tool for all of you to look at today. So this is, just imagine that you were the farmer and that you received this in the mail, and I'll just pass it around and you're feel free to take a look at it so you can get an idea of how this all works, and maybe you're interested in writing survey tools yourself. So I'm going to talk a little bit about the development of the survey tool. So the survey evaluated the farming practices of the farmer. So we were interested in farming practices like no tillage, use of cover cropping, etc. We also evaluated their demographics, such as their education, their income, things like that. Farm characteristics, time and organic management, the area of their farm are some of the variables that we collected. And then it's really important for you to understand that when we're talking about what someone believes, what I'm talking about is what farmers think about the microbiome and how their farming practices or other factors might be influencing the microbiome within their farming system. So these are kind of the belief variables here in this importance of category. So we asked farmers, do you think no tillage is influencing the microbiome within your farming system? Do you think climate change is influencing the microbiome within your farming system? These types of questions, and then they ranked how much they believed in those causal factors. We also evaluated their motivations to adopt a new practice discovered by the project to support the microbiome in their farm. And those motivations spanned from economic reasons to pest suppression reasons interactions with extension agents or their peers, as well as outcomes for biodiversity. So the survey also included a soil sampling kit, which is the part of the project that's going to allow me to build this, what I call the static socio-ecological model, which relates what they believe to their practices reported in the survey tool, and then ultimately the ecological outcomes that we derive from the soil. And I'm going to be talking a little bit about this later. So in the survey packet, they received a sampling bag and instructions on how to do that. So this survey underwent extensive pre-testing from 20 extension agents and farmers throughout the state of New York. And it's important to remember that, that these survey tools, they are essentially perceived by who receives them. And so when you send someone a survey tool, they have to understand what you're asking them. They have to understand that you're asking them about a specific practice. For example, no tillage. You have to use the language of a farmer so that they select the correct option. Otherwise, if they might say, "Oh no, I'm not doing that," then you're missing out on critical data that you would have been able to collect if you'd asked the right questions in the right way. So I want to just thank all of the folks who are extremely helpful in developing the survey tool, including our very own Meg McGrath, who is up there. I think she's next to that super large pumpkin. Okay, so imagine you're the farmer. I sent you this, well, actually what I did first was I sent a recruitment letter to the 280 farmers that I identified to meet our selection criteria. And this letter said, we're going to do this survey. We really want you to participate. If you don't want to receive the survey, let us know. One farmer called me and said, I don't want to be a part of your survey. Take me off your list. So ultimately, the surveys were sent to 279 farmers that met our selection criteria. After a short period of time, we also sent them a reminder postcard, and this postcard went to non-respondents, so people that I hadn't received soil samples from yet received this first reminder, and then they also received, non-respondents received a second reminder, and I did a targeted telephone solicitation to farmers in counties that I hadn't heard from yet to try to promote participation in the project. So the total sampling period for the project was 77 days. So I want to talk a little bit about the statistics now. I've kind of talked about the nuts and bolts of the survey process. So to be able to understand how farmers vary in their beliefs, it requires some collapsing of the data that we collected. So for the data that we collected on farmers' beliefs in the microbiome, what farmers think are influencing the microbiome within their farming system, I conducted a principal components analysis that collapsed the dimensionality of those questions down to 2 dimensions. And then I conducted affinity propagation, which is another clustering approach, to ultimately develop clusters that represent the beliefs of farmers. So any farmer that falls within a cluster ultimately held similar beliefs in terms of the factors that influence the microbiome on their farm. And then to verify that the clustering solution was optimal, I also evaluated, I also varied this thing called the preference value, which evaluates convergence and makes sure that your clustering is the optimal solution. So then I'll talk a little bit about the regressions and model selections that are ultimately going to tell us what mediates production practices. So as you might guess, what I'm modeling here is production practices as a response variable. So the production practices we evaluated included irrigation, cover cropping, fertilizer use, crop diversity, tillage, mulching, pre-planting, and pesticide use. And then I also included a variable for motivations. So as a dependent variable, which evaluates based on the other factors, the explanatory variables, how motivated was someone to adopt a practice to help promote the microbiome within their farming system. I also wanted to mention that this motivation data, I wanted to look at individual motivations, and so I conducted some Wilcoxon rank sum tests to evaluate how individual variables motivate the farmers within our survey. And so then the explanatory variables for this modeling approach include the belief clusters. So what does someone believe and how does that influence what they do, their production practices? Farmer demographics, like I said, variables like age, generation, education, and income. And then farm characteristics, variables like area, certified area and organic production, and time and organic, sorry, the amount of time the farm had been certified organic. I also included a couple of extra terms in this modeling approach or this model selection approach to get at just some basic like data control things. So there was an effort variable, and then there was also null models that were included in our modeling approach. Finally, I wanted to talk a little bit about the intercept in these models. So for models that we're looking at the clusters of farmer beliefs, what you'll see in the coming slides is that there's a cluster at the center of the principal component analysis. And that cluster is serving as the intercept in models that evaluate the response of production practices or motivations to farmers' beliefs. And so all comparisons are from that central cluster of farmers. Okay, so I'm gonna go ahead and get into some of the results here. The survey generated a 30.5% response rate. This was one farmer more than we desired. So a 30, which is good. The target for us was about a 30% response rate, and usually surveys don't ever get to the desired response rate. So 30% is like the minimum threshold for a response rate that you would want in a survey, but most of the time you don't get there. So this shows that we had a lot of engagement from the organic farming community. So 85 farmers responded to our survey, so 85 out of 279. And then this is a map of where we received responses from. Red are counties that didn't respond to us, but had farmers that met our selection criteria. Blue are counties that did not have any certified organic farms meeting our selection criteria. And then any other color represents counties that we received surveys from, and then the numerical counts from each of those counties. So you can see Long Island had 11 surveys submitted. We had really great support from extension people on Long Island in addition to Meg. So big shout out to Meg again for helping with generating interest in the survey tool. But you can also see that participation was incredibly widespread throughout the state. Okay, so I want to talk about the clustering of farmers by their beliefs. We determined that a 7 cluster solution was the optimal solution. So this was when I modified that preference value. So this just shows you that there are a lot of unstable clustering solutions. So these are unsupported solutions. And then this long straight segment are a series of 7 cluster solutions that was highly stable, and we selected that as our optimal solution for the number of clusters that we could support in terms of grouping farmers by their beliefs of the microbiome. So this is the cluster analysis reprojected into the principal component space. So what you can see are the 7 clusters here. Each cluster is represented by a color. You can see incredibly good separation of the clusters. And then the 6 main clusters exist kind of in this, sorry, I'm gesturing with my hand, in this space here. And then we have the 7th cluster down here in the bottom left-hand corner. So I want to explain each of the axes here. So on this rotated component 1, these are factors that are external to the farm. So these are farmers that either believe factors external to the farm, like climate change and pesticide use in bordering lands influences the microbiome within their farming system, or for example, didn't believe that. And then on the x-axis here for RC3, we have on-farm management. So these are farmers that believe things that they're doing within their farming system either influence the microbiome within their farm, or don't influence the microbiome within their farm. So typically, what you see is that farmers on the negative component, negative part of each of the axes are farmers that generally did not think that either external factors or factors internal to their farm are influencing their microbiome. And then this is the central cluster I was talking about earlier. This is the intercept in the models. Does anyone want to guess what this cluster, what their response was? It's really common in survey tools. But it's similar to no answer, but these are all the farmers that said everything was somewhat important for influencing the microbiome within their farming system. So this is just the middle of the road people who are just like all the way down the survey. They were like, everything is just a little bit important. This is really super common in survey tools where people are like, I'm just not sure. So I'm just going to put somewhat important all the way down. So I'm not going to spend a whole lot of time on all of the results, but I just wanted to show you that from our modeling approach, we did find that what farmers believe does influence or is correlated to the practices that they're using. And then farmers that, for example, believe that their farming practices influence the microbiome ultimately use fewer practices that we think could be detrimental to the microbiome, like pesticide use. So I'm not going to walk through all of these results. I don't have enough time, but I also wanted to show that a lot of the farming practices were also related to farm characteristics and demographics. So farms are complex socio-ecological systems, not only their beliefs that are regulating the practices that they use. So a few of the factors that we found were important for influencing practices that we think could mediate the microbiome include the farmer's age and their source of income, as well as their certified area in organic production and time in organic production. So you can see that, for example, a lot of these trends are kind of like downward facing. So for example, one of the things that we think could be related to microbiome diversity within organic farming systems is crop diversity. And what we found is that as farms have more certified acres in organic production, they actually reduce their crop diversity. So that might ultimately tell us that some of these larger farms that are less diversified might have simpler microbiomes, but we don't know. We can test that. And then finally, I wanted to talk a little bit about, we've talked a little bit about like what's influencing farmers' current farming practices or their adoption of practices so far, but I wanted to talk a little bit about what might motivate a farmer to adopt a new practice to support the microbiome. So what we found is that farmers that tended to believe either factors external to the farm or their farming practices are important for mediating the microbiome within their farm were more motivated to adopt a new practice. So farmers that are already kind of interested and cued into the factors that are mediating the microbiome within their farming system are also more motivated to adopt a new practice. And in addition to that, motivations were strongly driven by pest reduction. So when you asked farmers, or when we asked farmers, what might motivate you to adopt a new practice to support the microbiome within your farm, pest reductions were the top factor for mediating their motivation. So I'm just gonna kind of wrap this up here by talking a little bit about some of the key take-home messages. This is some data that I didn't go through earlier, but we also asked farmers, do you believe that the microbiome within your farm can enhance pest suppression through mediating plant defenses? And they said yes, they believe in what we're studying here. So that's really super important because we want the farmers to at least be interested in and believe in this process as kind of square one. We did find that beliefs mediate practices and farmer motivations, and that farming system characteristics also mediate farming practices that could influence the microbiome. So you don't always have to know what someone believes to understand their farming practices. And ultimately, some of these data are widely collected by the USDA and might be able to make some predictions about farming practices that farmers are using at broad geographic scales. And And then if we understand which farming practices are related to microbiome conservation, then we might be able to get sort of an idea or prediction of where farming practices are being used that might support the microbiome, or the opposite of that as well. So this is kind of interesting. When we think about what farmers believe, some farmers didn't believe that their farming practices influence the microbiome within their farm. So when we're thinking about shaping and educating and doing outreach, it's really important to understand where farmers are starting from in terms of their knowledge. And so we can start addressing what farmers think about the microbiome, and extension efforts can be geared towards addressing farmer beliefs and their pedagogical approaches. And then the final key take-home message was that proof of pest reductions is an important part of motivating farmers to adopt a new practice to support the microbiome. But several of these factors are also very highly ranked together, like ease of integration, environmental benefits like conservation and increased yield. So really any one of those, if we could prove any one of those, that might help motivate farmers to adopt a practice to support the microbiome within their farming system. Okay, I really sped through that because I wanted to get to the kind of ecological component of this model. And I'm tentatively titling this, eventual paper, as Farmer Beliefs Indirectly Mediate Microbiome Diversity, Destabling Pest Populations in Organic Farming Systems. The original paper that Claire wrote on the subject went to Nature Plants. So I'm thinking, maybe this will go to Nature Plants as like the follow-up to Claire's study. I think we could maybe shoot for that. I don't know. We'll see how it all turns out. So as you'll recall, the survey tool also included instructions for how to submit a soil sample. So we asked farmers to collect some like very basic equipment from their farm, like a bucket and a shovel and a knife. Then we told them we wanted them to go out to their field and evenly sample the field, avoiding any strange areas. And then basically we just asked them to take a soil sample core with their shovel. So you can see we asked them to kind of cut away at this excess here and have like about a 6-inch core. And then we kind of had them mix all of the subsamples together in that bin. And so it's a bulk soil sample from their field. So it's important to note that this soil sample came from the field where they recorded their production practices. So this soil sample is a representation of the ecological outcomes that they recorded in their survey tool. So they sent us the survey tool back, and those also came with the soil samples. So we ended up with a bunch of soil samples from these farmers. It was really cool. If you ever want to work with mail services on something like that, there's a lot of details to making this happen, but it's a really, really neat. So upon receiving the samples, the soil went into 2 different pipelines. The first pipeline was the bioassay pipeline. So the soil sample was stored in a walk-in cooler, and then I used parts of that soil sample to run bioassays. And then another part of that soil sample went into the sequencing pipeline, where I took a portion of the soil sample and I stored it immediately in a -20 freezer. And then I took soil out of that soil sample for sequencing of the soil microbiome. So I'll talk a little bit about the sequencing pipeline. I think, for people in this department, this pipeline is incredibly familiar and it's, a cakewalk for you. But for me, I came to learn kind of about this kind of stuff. So as part of the point of me being here. So the first thing was that I stored it in the -20. For this pipeline and for this kit, it needs to be super dry. So I dried the soil in the BSC. And then I just ran it through a kind of typical soil DNA extraction kit from Quigen. I used PCR to verify that the target region's amplified. And then I sent the the DNA samples out for each of the soil samples for sequencing of the V4, V5, and ITS region at Dalhousie University. So I didn't do the sequencing or library prep or any of that stuff in-house. And then the results came back and I ran the files through the Data2 pipeline and I assigned taxonomy using the Silva and Unite community databases. I'm really not going to talk at all about taxonomy assignment today. And everything that I'm going to show you today is at the level of the ASV. So that kind of gives you an indication at the resolution that I'm working with here. So once you have this community data, you need to kind of develop some measures for it. And so one of the thoughts is that I would use hill numbers to quantify different aspects of diversity or biodiversity within each of the soil samples. And so I've done this, but today I'm only going to focus on the Shannon index. And the Shannon index is an index that accounts for the relative abundance of each of the ASVs. And then some of these other measures like richness is just a count of each ASV, for example, and Simpson's index accounts for unevenness within the community matrix. But I'm just going to talk about Shannon index, which is includes the relative abundance of each of the ASVs. So that's the sequencing pipeline. I'm going to talk a little bit about the bioassay pipeline now. So these are the soil samples that went into the walk-in freezer. One thing that I haven't explained to you yet is that within this clustering approach, one of the values of using affinity propagation is that it identifies exemplars. So each of these square boxes are real world farms that represent the beliefs of all the other farms within that cluster. So rather than using all 85 farms to evaluate the ecological outcome of the soil sample on pest suppression, I'm able to just use the 7 exemplars represented by these points in those bioassays, which you can imagine greatly reduced the amount of replications and things like that that I needed to do. So the exemplar soil samples, we use an extraction method that's very rudimentary. It's the whole bulk soil sample. We take some of that soil, we put it into a nutrient solution and we shake it together. And then we try to get as much of the soil out. So what's left in the nutrient solution is just what we think is a representation of the microbiome within that soil sample. So then I took that solution, that nutrient solution, and I applied it to pea plants. I put aphids on those pea plants, and then I counted the progeny of those aphids after 9 days. And then I also took leaf tissue samples to quantify plant defenses. So this is another representation of my bioassays. I'm just showing this to kind of give you an idea that half of the plants received aphids and half of them did not receive aphids. And so this other half here will allow us to evaluate eventually priming via the microbiome. And then the other half of this will be able to kind of look more at like induction or recruitment. So what happens when you apply an aphid, and is the plant able to recruit the microbiome to defend itself after the aphid has been applied? So some additional controlling covariates here. So we know that, for example, farming practices might mediate the microbiome, but soil properties are incredibly important. So I used the USGS Web Soil Survey, and each one of our farms, I extracted data on pH, organic matter, percent sand, silt, and clay, available water content in this variable called the erosion factor. And so all of this data was also pulled down to help control for how soil properties could be influencing the microbiome and not other things like farming practices that we're interested in. So how do you control for those variables? That's what we're doing here. This web soil survey is great. The resolution, the spatial resolution is incredibly fine. And if you're good at GIS, it's a great resource. Okay, so you might be asking yourself, how are you going to model all this information together? And so what Claire and our collaborator ultimately decided on was using a structural equation modeling approach. And so best practice is defining what your meta model is. And so this is a meta model of how we think, how we theorize the system works. So the basic idea is that beliefs mediate farming practices. Those farming practices mediate microbiome diversity. Microbiome diversity ultimately mediates plant defenses. Plant defenses ultimately mediate pest suppression. This is how we think the model works. But we also want to control for other things like farming system characteristics like time and organic management, which I hypothesize could have a direct effect on microbiome diversity, or could influence farming practices, which then indirectly, time and organic management would ultimately indirectly impact microbiome diversity. Demographics, we don't think those have a direct impact on the microbiome, but again, that would be mediated through farming practices. And then those abiotic factors, the soil properties, we think those could have a direct influence on the microbiome, but also an indirect influence through farming practices. So I'm going to show you some kind of complicated models in a second, but I wanted to just let you know that just like any modeling approach, you can over parameterize the model and then you get really bad estimates. And so for this modeling approach that I'm going to tell you about, I kind of used my inference to select some of the variables that I thought might be important, either to science or to farmers. So for my abiotic factor, I picked organic matter because the farmers said in their survey that they thought organic matter was an important factor for influencing the microbiome within their farming system. So I wanted to test that. In terms of farming practices, you saw from my survey that there's a lot of different farming practices that I evaluated, but not all of them are farming practices that if you told a farmer to adopt, they might be like, hey, I'm going to do that tomorrow. So I decided to focus on two farming practices. One is no tillage, which is increasingly adopted within organic vegetable production, but it's still on the adoption curve. Not everyone is doing it. And then pre-planting practices like solarization and tarping, which is also gaining traction in New York State. So also included in the model were the clusters, so their beliefs. For my demographical variable, I picked income, which we found was an important variable for influencing the adoption of farming practices. And then for farm characteristics, like I said before, I picked time and organic management. And also thinking back on this, I might have included a couple of other farming practices in here, like the application of compost was one, and the use of legume cover crops. So all of these are practices that I think farmers might reasonably adopt if you said, hey, this is going to really help microbiome and pest suppression within your farm. How am I doing on time? Almost out of time. There's some theory. I'm going to skip over this, and I'll just show you the model. So what I found in this model is beliefs do influence, indirectly influence the bacteriome or bacterial diversity, and this influence was caused through no tillage. So we don't believe that beliefs influence the bacteriome directly, but what we found is that as farmers tended to believe more that their practices or external factors influence the microbiome within their farm, those farmers tended to adopt no tillage or use no tillage. And as farmers adopted no tillage, this increased bacterial biodiversity within their soil sample. And then this is the model with my bioassay data in it. So the previous one was just the sequencing data. And what I found here is that there's indirect links between beliefs and variation in the aphid population. And so what I ultimately determined is that beliefs indirectly influence pest outbreaks or variation in the aphid population through no tillage and bacterial biodiversity. So key take-home points here: farmers' beliefs, they tend to promote practices that enhance the microbiome and perhaps stabilize pest populations. So reduce the variation in their reduce the variation in the population and keep it from reaching outbreak levels. And these outcomes were mediated by no tillage and the bacteriome. And that just the final take-home message here is that stable pest populations may be less inclined to outbreaks and easier to suppress for farmers. So I still have to include the plant defense data in this model, so it's not a finished model yet. And then the final step in this project is to create what's called a dynamic socio-ecological agent-based model, We're going to extend this information back to farmers and see if they're interested in adopting any of these practices to help promote the microbiome and pest suppression within their farm. So that's it. Thank you for your time. Any questions or comments? And if you do social media, you can find me on Twitter @sustainability. And that's it. So I have a question in the chat. The chat question is, which pre-planting practices reduced fungal diversity? So yeah, so this is a really interesting outcome here. We're talking about, oops, can we not go back? Okay. We're talking about this pathway here. And so we're talking about solarization and tarping here. Those were grouped together into one pre-planting practice variable. And we were really interested in this pathway too. And what we think is going on is that pre-planting practices like solarization and tarping are reducing fungal diversity, but it's possible that those reductions in fungal diversity could be the reduction of pathogenic species. And we just haven't done the follow-up analysis to understand this. Because if you look at this, the addition here, we do see that the remaining fungal diversity also has this influence on the aphid populations. But we also, I've done a little bit of analysis that does the modeling on a species by species level. And some of the top species that are lost due to pre-planting practices are potentially plant pathogens. And that's one of the reasons that you would want to use those practices. So there's definitely a lot of follow-up analyses that we want to do with these data.
I think we're out of time. So join me again. Thank you, Eli.
This has been a production of Cornell University, on the web at cornell.edu.